Hello everyone. Today we are here to discuss about the topic precipitation. Here we go with introduction, precipitation reactions in fluids, precipitation reaction in gels, radial immunodiffusion, double immunodiffusion and conclusion. Here we go with introduction. Precipitation in immune techniques is a method used to detect and quantify antigens or antibodies based on their ability to form insoluble complexes. When a specific antigen and its corresponding antibody interact in optimal proportions, they create a lactide structure that precipitates out of solution. This process is fundamental in various immunoassays, including immunodiffusion and immunoelectrophoresis. Precipitation reactions can be performed in liquid or gel media and are useful for analyzing protein concentration, purity, and identity. The sensitivity and specificity of these techniques make them valuable in diagnostic laboratories for detecting infections, autoimmune diseases, and other conditions. By observing the formation of precipitates, scientists can interfere the presence and concentration of specific proteins, providing crucial insights into immune responses and disease mechanisms. Precipitation reactions. Precipitation results from the interaction of a soluble antibody with a soluble antigen to form an insoluble complex. Antibodies that thus aggregate soluble antigen are called precipitants. Formations of an AgAb lactase depends on the valency of both antibody and antigen. The antibody must be bivalent for precipitation reaction. Monovalent fragments cannot form precipitate with antigen. Similarly, the antigen must be either bivalent or polyvalent, that is, it must have at least two copies of the same epitope or have different epitopes that react with different antibodies. Precipitation reactions in fluids. Precipitation occurs maximally only when there are optimal proportions of the two reacting substances, antigen and antibody. Hence, an insoluble antigen antibody complex formation occurs within a narrow concentration range known as the zone of equivalence. This represents the condition under which macromolecular antigen or antibody complexes are formed that are sufficiently large to be precipitated. Outside the equivalence concentration, conditions known as antigen or antibody excess occur, which result in the formation of small soluble complexes. When increasing concentrations of antigen and added to a series of tubes that contain a constant concentration of antibodies, variable amounts of precipitate form if the amount of the precipitate is plotted against the amount of antigen added. A precipitant curve is obtained. Precipitation in gels. Precipitation reactions carried out in agar gels are referred to as immunodiffusion reactions. When Ag and Ab diffuse toward one another in agar, or when Ab is incorporated into the agar and Ag diffuses into the Ab containing matrix, a visible line of precipitation will form. Visible precipitation occurs in the region of equivalence. No visible precipitate forms in region of antibody excess or Ag excess. Two types of immunodiffusion reaction can be used to determine the relative concentrations of antibodies or antigens as well as the identity of antigens. These immunodiffusion techniques are radial immunodiffusion and double immunodiffusion. Radial immunodiffusion or Mancini method. The relative concentration of an Ag can be determined by a simple quantitative assay in which an AG sample is placed in a well and allowed to diffuse into agar containing a suitable dilution of an anti-serum. At the region of equivalence, a precipitation ring forms around the well, the area which is proportional to the concentration of Ag. By comparing the area of the precipitation ring with the standard curve, the concentration of the antigen sample can be determined. Double immunodiffusion or Ucherloni method. The Ucherloni method, both AG and AB diffuse radially from wells towards each other, thereby establishing a concentration gradient. It is an effective quantitative tool for determining the relationship between AG and the number of different AG-AB system present. Applications of precipitation, diagnosis of diseases, quantification of proteins, vaccine development, purification of antibodies, and immunoelectrophoresis. Conclusion. Precipitation techniques are fundamental in immunology for detecting and quantifying antigens and antibodies. They provide a simple yet effective method to study immune responses, diagnose diseases, and quantify proteins. Techniques like immunodiffusion and immunoelectrophoresis leverage precipitation to provide valuable insights into antigen antibody interactions, making them crucial in clinical diagnostics and research. Their ability to purify antibodies and analyze complex biological samples further emphasizes their importance. Despite newer methods like ELISA and flow cytometry, precipitation remains a reliable, cost effective tool in immunotechnics, highlighting its enduring relevance in the field of immunology and biotechnology. Thank you.